Well, now here, eukaryotes, for the most part, are well behaved. A eukaryote makes an mRNA, it goes to the ribosome, the mRNA makes your protein, just like I taught you. But now prokaryotes are a little weird. Prokaryotes you know, their messages are fine. None of this funny cap business, none of these poly A tails, none of this splicing. But here's the weirdness that prokes do. This mRNA, one mRNA might encode multiple different proteins. Multiple proteins. This is one protein. This, multiple proteins. So what happens is, this RNA, the ribosome gets on here at a certain site. Now remember I told you the ribosome finds the first AUG? Well, it's a little more complicated. There's a particular ribosome binding site that ribosomes like to go to. It turns out that this prokaryotic message has several of those ribosome binding sites. Ribosomes sit down. They then scan for the first AUG that they see after that point and start making a protein. So I might have several different proteins all being made by one messenger RNA. One RNA making multiple proteins. Why would I do that? Why not just like, wouldn't it be simpler to make your head hurt less to have one gene making one protein instead of these separate sequences encode, this one mRNA encoding multiple proteins? So they're related. Tell me about that. If they had similar functions, it might be very efficient to have regulatory controls that made one message, and then I get all the proteins made together, rather than having separate regulatory controls for each message. Suppose I'm a prokaryote and I'm dividing very rapidly and I care about having a small genome. It might be very efficient to do it this way. So what related genes might you put together on the same message? Same pathway. Maybe, if you're in charge of the committee, you would say, I would like to arrange the genes encoding the multiple steps of a biochemical pathway. And that's often what happens. It's exactly what happens. So you can get co-regulation of genes. Of genes in the same pathway. And such a thing, this, which is called a polycistronic message. Polycistronic is an unhelpful name for it, but it's what they call it sometimes. Or an operon. A regulatory thing that makes many such things. Now, why do bacteria do it? To minimize DNA. They just want to use as little space as possible. So once it's invented a regulatory control that turns on this message when I want to make arginine, well, just stick all the genes on the same message. It's cheaper, simpler, less DNA needed. Now, who really has trouble with the amount of DNA they can have? You know, do you have trouble with I mean, is replicating your DNA a rate-limiting step in your replication as an organism? Now, what is the rate-limiting step in your organismal replication? You're having offspring. Sorry? It's graduating from MIT, getting a job, things like that, right? And DNA replication is not rate limiting, actually, right? Getting a degree, you know, all that, that's rate limiting, right? But for bacteria, who are dividing not every, you know, reproducing not every 20 years, but every 20 minutes, DNA replication is a rate limiting step. It's important to their replication, and they want compact genomes. You don't care about a compact genome. It's simply not a big metabolic cost to you. Viruses, think about viruses. Their genomes are tiny. They're much tinier even than prokaryotes. Typical viruses might have a few thousand or tens of thousands of bases. Viruses were millions of bases. Sorry, bacteria were millions of bases. Viruses sometimes might only have 10,000 letters of nucleic acid. They really have to use their information very compactly. So you know what viruses do sometimes? Not all, but some. Some viruses make a messenger RNA. They have a ribosome binding site right here, and they start translating the message. Let's give it a sequence. A, C, U, T, G, U, G, A, G, C, A, and we'll put an A, U, G in front of that. They could start translating here. Whoops, I'm just going to fix that. But you know what? There's also an AUG over here. They could start using that one. And you know what else? If they found another AUG off frame, some of them use that too. Remember, the first AUG in normal messages gets used to set the phase of the codons. But in theory, I could be reading those codons in a different reading frame, shifted over by 1 or shifted over by 2. Some viruses are so clever that they encode 3 different proteins smack on top of each other by reading the same nucleic acid sequence, it shifted 3 different ways. It makes your head hurt to think, how could I make a functional protein? I've got to manage to get 3 separate functional proteins built out of a single nucleic acid sequence. You know, one, it's easy. Just tell me the amino acids. Now this becomes an interesting puzzle. Can I make a sequence where read 1 out of frame, it also makes a protein I want, and another one where, and viruses have evolved, some viruses have evolved to do that, showing you how much they care about economizing their DNA. All right, so what do we got? We have DNA replicates. We've seen how it replicates. It's mostly the same by nucleic acid copying, but all these variations, linears, circulars, single strand, double strand, RNA, reverse transcriptase, transcription, translation, look over these examples, and what they should do is help solidify how these things work and how they're used in slightly different fashions. Next time.